Start minute.
Good morning, students. Welcome to another session of molecular biology. This is Sarala, JL in Botany, working at Hasanparthy, Warangal. Welcome to Telangana Residential Educational Institution Societies for webinar online classes. So, in the previous class, I had given assignment. So, some students done their work very well. Divya, Nandini, Shravani, and Chandana from Balnagar. Maunika, Deepthi, Sadaf, Varsha, and Akila from Vaira. These students, they done the assignment very well. All the best, students. So, in the previous class, we had already discussed about structure of DNA as well as replication of DNA and types of RNA also completed. Then today we have to discuss about transcription. Transcription. What is transcription? Transcription means the process of copying genetic information from one strand of DNA into RNA. Transcription. Transcription means the process of production or formation of RNA from DNA is called as transcription. Listen carefully. This transcription as well as translation process both are very, very important for your need point of view. Transcription means the process of copying genetic information from one strand of DNA into RNA. Which means the synthesis of RNA from DNA is called as transcription. DNA molecule two strands. Out of two strands, only one strand of DNA is involved in transcription process to produce the RNA molecule. This phenomenon is called as transcription. Transcription process takes place both in prokaryotes as well as eukaryotes. So when we observe the transcription unit in the DNA, so first all of you understood the definition or transcription. Transcription means synthesis of RNA from DNA is called as transcription. So a transcription unit in the DNA consists three regions: a promoter region and the structural genes and the terminator region. Here listen carefully. The transcription unit in the DNA consists three regions: a promoter region and the structural genes and a terminator region. So it is a transcription unit. So these two strands are DNA strands, and this strand is runs in 3' to 5' direction, and this strand is runs in 5' to 3' direction. So these two strands are DNA strands, and this strand is runs in 3' to 5' direction, and the another strand is runs in 5' to 3' direction. Okay. So out of these two strands, only one strand is involved in transcription process. That strand is template strand. That strand is called as template strand, and this template strand is involved in transcription process. And this strand, another strand which is present in the DNA molecule is called as coding strand. which does not involve in the transcription process. Is it clear, all of you? These two strands are DNA strands. And this DNA, in these two strands, only one strand is involved in the transcription process. That strand is called as template strand. The other strand, which does not involve in the transcription process, and this strand is called as coding strand. Okay, so when we observe the transcriptional unit, it consists three regions. So a promoter region, this is the promoter region, and structural genes and a terminator region. These three parts are three regions are present in the transcriptional unit. Okay, this promoter region, which is present at the 5' end of the coding strand, and the terminator region is present at the 3' end of the coding strand. And the structural genes, they are located at the middle portion of the coding strand. There is little bit messy or confusion is there. So students, listen carefully. This promoter, which is present at the 5', there is a doubt arise for you. Only template strand is involved in the transcription process. Coding strand does not involved in the transcription process. So this promoter region or promoter is present at the 5' end of the coding strand, which is located in the template strand. Okay, and a terminator, which is present at the 3' end to the coding strand on the template strand. These two strands are there, and only this template strand is involved in the transcription process. Promoter is located towards the 5' end of the structural gene, and the terminator is located towards the 3' end of the structural genes. Structural genes are present towards the middle portion, and this promoter is present at the 5' end of the structural gene, and the terminator is located at the 3' end of the structural genes. And these three regions are present in the transcription unit. And out of these two DNA strands, only one strand is involved in the transcription process. That strand is called as template strand. There is a doubt arise for you people. DNA consists of two strands. Why two strands of DNA are involved in transcription process? For example, if two strands of DNA are involved in the transcription process, and the two strands of RNA or RNA molecule is synthesized. In a previous class, we already discussed about RNA. RNA is a single stranded molecule. It does not consist two strands. It consists only RNA molecule consists only one single strand. It does not consist two strands. But this DNA, it consists two strands. Out of two strands, only one strand is involved in the transcription process. Another strand, that is coding strand, which is not involved in the transcription process, because RNA is a single stranded molecule which is synthesized from this template strand. If these two strands are involved in the transcription, two strands of RNA are synthesized, and there is a great confusion and mistake is developed in the particular organism. It leads to the mutations in the particular organism. Is it clear? So that's why only one strand, that is template strand, is involved in the transcription process. The strand that act as the polarity, that is 3' to 5', acts as template, and it is also referred as template strand. This strand is the template strand which having the polarity towards 3' to 5' direction, and it is called as template strand. The other strand which has the polarity towards 5' to 3' and the sequence same as in the RNA is displaced during transcription. This strand is called as coding strand. Is it clear? The strand which has the polarity towards 5' to 3' end, which is same as the sequence of newly synthesized RNA, is called as coding strand. The newly synthesized RNA is, the synthesis of RNA is takes place in this manner towards 5' to 3' direction, and this newly synthesized RNA is having same basis to the coding strand. Is it clear? So only one strand is involved in the transcription process. This is called as template strand. And the newly synthesized RNA is having the polarity towards 5' to 3' direction. Okay. The other strand which has the polarity towards 5' to 3' direction and the sequence same as RNA in displaced during the transcription. This strand is called as coding strand. So the process of transcription is consist 3 stages: initiation, elongation, and termination. The process of transcription is completed through 3 major steps: initiation, elongation, and termination. So the total process is completed through 3 steps. First step is initiation. Before this initiation, we already know about the nitrogen bases which are present in the DNA. There are 2 types of nitrogen bases. They are purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are thiamine and cytosine. So, a purine always pairs with a pyrimidine. A pyrimidine always pairs with a purine. In the case of RNA molecule, in the place of thiamine, uracil is present. That uracil is pairs with adenine. So it is the difference in between DNA as well as newly synthesized RNA, nothing but mRNA, messenger RNA. So this transcription process, it consists of 3 stages. First stage is initiation. Initiation. In this step, the RNA polymerase enzyme, this enzyme, RNA polymerase enzyme, plays an important role in total transcription process. This RNA polymerase enzyme is also called as transcriptase. DNA dependent RNA polymerase or transcriptase. This enzyme binds to the promoter site on the DNA and it unzips or unwind the DNA double helix. Is it clear, all of you? In this initiation process, RNA polymerase enzyme. In this total transcription process, RNA polymerase enzyme plays a vital role. This RNA polymerase enzyme is also called as DNA dependent RNA polymerase or transcriptase. First, this enzyme is bind to the promoter site. This is the promoter site. So at this promoter site, RNA polymerase enzyme is bind to initiate the transcription process. In this initiation, there is a factor that is nothing but initiation factor, which is plays a vital role along with RNA polymerase enzyme. Okay, this RNA polymerase enzyme bind to the promoter site on the DNA and it unwind or unzips the double helix because RNA molecule is present in double helical structure. So in this transcription process, first this RNA polymerase enzyme unzip or unwind the DNA double helix to initiate the transcription process. Next, second step is elongation. Elongation means that RNA formation is takes place continuously and polynucleotides are synthesized continuously and polymerization process is takes place. Polymerization process means the genetic information which is present on the DNA is copied into the RNA molecule. Okay, so in this elongation, free nucleotides bind to their complementary pairs on the template strand of the DNA and elongating the RNA chain. This RNA chain is identical to the informational strand of the DNA except the nucleotide thymine in the DNA. Is it clear? In the DNA, thymine is present. But in the case of this RNA, thymine is replaced by uracil. And the polymerase enzyme moves along the DNA in 3' to 5' direction and extending the RNA in 5' to 3' direction. Dear students, all are remember a important point, that is transcription process is always takes place in 5' to 3' direction. Why it is always takes place in 5' to 3' direction? Means the DNA strand which having the polarity towards 3' to 5' direction, which is acts as template strand and which is involved in the transcription process. So that's why the transcription process is takes place in 5' to 3' direction and the RNA synthesis is takes place in 5' to 3' direction. The last step in the transcription process is termination. Termination means there is a ending for the polymerization of RNA from DNA. It's the specific sequence in the DNA signals termination of transcription. A specific sequences which are present in the DNA, they are TUS elements and TER proteins which are present in the DNA which gives the signals for the termination factor. There is a initiation factor and there is a termination factor. Both are involved in the transcription process. So This is the DNA molecule, which is runs in 3 prime to 5 prime direction. This is the newly synthesized RNA. This RNA synthesis is takes place 5 prime to 3 prime direction. So in this DNA, here is promoter region and here is terminator region. The middle portion, structural genes are there. So in this termination process, the terminating this is promoter region. This is the promoter region and this is the terminator region. This one is promoter region, this one is the terminator region on the DNA. It says specific sequences in the DNA signals the termination of transcription. Specific signals means ter proteins and tusk elements which are present at the terminator region. They are initiates for the they involved in the termination process and they can stops the transcription. Sorry for the interruption. My mobile is stuck suddenly. జావేద్ సార్ స్క్రీన్ షేర్ ఆగిపోయింది సార్. జావేద్ సార్, కాశీనాథ్ సార్. స్క్రీన్ షేరింగ్ స్టాప్ అయిపోయింది. జావేద్ సార్, కాశీనాథ్ సార్, జావేద్ సార్, sorry children, sorry for the disturbance. So it is about termination process. So in total transcription is completed through 3 steps: initiation, elongation, and termination. So this transcription process, this is a double-stranded DNA molecule, and at the process, at the time of transcription, these 2 strands are separated from each other, and that unzip is caused by RNA polymerase enzyme, and this strand is the coding strand which does not involved in the transcription process, and this strand is template strand which is involved in the transcription process, and this template strand is runs in 3' to 5' direction, and this is the newly synthesized RNA, and this total transcription process is catalyzed by RNA polymerase enzyme, and this RNA is synthesized towards the this RNA molecule synthesis is takes place 5' to 3' direction, because template strand is runs in 3' to 5' direction. Once this RNA synthesis is completed and this RNA molecule is transfers from nucleus to the cytoplasm region. Is it clear, all of you? This DNA molecule is present inside the nucleus. So that's why this transcription process is takes place inside the nucleus. Okay. And RNA polymerase enzyme is associate transiently with initiation factor and the termination factor to initiates the initiation and terminates the transcription. This RNA polymerase enzyme, it is transiently attached. Transiently means temporarily attached with initiation factor to initiate the transcription, and attached to the termination factor to terminate the total transcription process. In the case of prokaryotes, especially in bacteria, transcription and translation takes place in the same compartments. Listen carefully. In bacteria, it is a prokaryote. In prokaryotic organisms, in prokaryotic organisms, in the place of nucleus, only genetic material DNA is present. It is not bounded by nuclear membrane. Okay, so that's why in prokaryotes, transcription as well as translation both are takes place simultaneously in the same compartment, that is nothing but cytoplasm region. In bacteria, transcription as well as translation, both are takes place in the same compartments. Means there is no well developed nucleus in bacteria. In the cytoplasm only genetic material DNA is present. So that's why these two processes are takes place in the same compartment. In this bacteria, many times translation can begin much before the mRNA is fully transcribed. Means before the completion of transcription, translation process is initiated in bacteria. Transcription as well as translation, these two steps are major steps in the protein synthesis process. Once DNA is synthesized, that DNA is involved in transcription process to synthesize the mRNA. That mRNA is involved in translation process to synthesize the proteins. So that's why these two steps, transcription and transcription as well as translation, these two are the major steps which are involved in the protein synthesis process. Okay. In the case of eukaryotes, there are two additional complexities. Eukaryotes means in eukaryotes, nucleus is bounded by nuclear membrane. And in this eukaryotes, in this transcription process, two additional complexities is there. There are at least three RNA polymerases in the nucleus. In the case of eukaryotes, there are three types of RNA polymerases are present in the nucleus because transcription is takes place inside the nucleus. In the case of eukaryotes, DNA genetic material is present inside the nucleus. So that's why transcription is takes place inside the nucleus. So that's why there are three types of RNA polymerases are present in the nucleus. The first one, RNA polymerase 1 transcribes the rRNA. RNA polymerase 1 transcribes rRNA. rRNA means ribosomal RNA. Next, second one, RNA polymerase 2, which transcribes the precursor of mRNA. Precursor of mRNA, that is nothing but hnRNA. Inside the nucleus, transcription processes takes place. But in eukaryotes, that mRNA is not synthesized completely. Incomplete or precursor of mRNA is synthesized in the nucleus. That mRNA is called as hnRNA, heterogeneous nuclear RNA. It is synthesized by the activity of RNA polymerase 2. Next, RNA polymerase 3 RNA polymerase 3. It transcribes the tRNA. tRNA means transfer RNA. So there are three types of RNA polymerases in eukaryotes. RNA polymerase 1, RNA polymerase 1, which transcribes the rRNA, ribosomal RNA. RNA polymerase 2, which transcribes the precursor of mRNA or hnRNA. RNA polymerase 3 transcribes the tRNA or transfer RNA. It is the first complexity. It is the first complexity in the case of eukaryotes. And the second complexity is that the primary transcript contains both exons and introns. Primary transcript nothing but hnRNA, heterogeneous nuclear RNA or immature mRNA. It consists both exons as well as introns. Exons are the coding regions and introns are the non-coding regions. Exons, it consists functional genes. Introns, it consists non-functional genes. So this primary transcript, it contains both exons as well as introns. So in this second complexity, when the introns are removed and the exons are joined or spliced together in a definite order, this process is known as splicing. Splicing means exons, introns are removed and the exons are joined together. This process is known as splicing. It is the most important event which is present in the eukaryotes. Then this hnRNA means primary transcript, means heterogeneous nuclear RNA. Why it is called as nuclear RNA? Means this RNA is synthesized inside the nucleus. Because DNA is present inside the nucleus, this RNA is synthesized inside the nucleus. So that's why it is called as heterogeneous nuclear RNA. It undergoes some additional processing called as capping and tailing. So this red color one is newly synthesized. This red color one is DNA molecule, and this green color one is RNA molecule. So from this DNA, RNA synthesis takes place. That process is called as transcription, and this is the nucleus. Inside the nucleus, the total process is takes place, and this RNA is hnRNA. It is separated from the DNA molecule. It undergo capping and tailing. Okay. Before that capping and tailing, it undergo splicing. Splicing means this introns. Introns means non-coding regions or non-coding sequences are removed, and exons are the coding regions, and functional regions are joined together. That process is called as splicing. RNA splicing. Splicing means introns are removed in a definite order and exons are joined with each other. That process is called as splicing. When once splicing is completed, capping as well as tailing both are takes place. Capping means at the 5 prime end of the hnRNA, a extra guanine molecule is added. Capping means an unusual nucleotide methyl guanosine triphosphate is added to the 5 prime end of the hnRNA. It is called as capping. At the 5 prime end of the hnRNA, unusual nucleotide. That unusual nucleotide means 5-methyl guanosine triphosphate, which is added 5-methyl methyl guanosine triphosphate. Here there are 3 P's. 3P means triphosphate. 5 methyl. M indicates for methyl. G guanosine. 3 P's. Triphosphate. An unusual nucleotide 5 methyl guanosine triphosphate is added at the 5' end of hnRNA. This process is called as capping. Is it clear? Then tailing. Tailing means at the 3' end of the hnRNA, adenylated residues are added. Adenylated residues. Polyadenylation means adenylated residues or adenine residues are added at the 3' end of the hnRNA. It is called as poly-A tailing, and the process is called as tailing. Tailing means at the 3' end of the hnRNA, adenine residues or adenylated residues are added. It is called as tailing. So once splicing is completed, capping and tailing is completed, and this is considered as the matured mRNA or messenger RNA. Once this post-transcriptional changes are completed, this matured mRNA is transferred from nucleus to cytoplasm in eukaryotes. In eukaryotes, the transcription process is much complicated when compared to prokaryotes. In prokaryotes, transcription as well as translation both are takes place in the same compartment, and the nucleus is present in the cytoplasm. There is no nuclear membrane, so that's why the genetic material DNA is present in the cytoplasm only. But in the case of eukaryotes, much complexities are there, and the RNA polymerase enzyme is 3 types. RNA polymerase, first complexity is RNA polymerase enzyme is 3 types. It is one RNA polymerase is specific for the synthesis of one specific type of RNA. And the second complexity is introns as well as exons are present on the hnRNA. Exons are coding sequences, they consist of functional genes. Introns are non-coding sequences, they consist non-functional genes. Introns are removed and exons are spliced or joined together. This process is called as splicing. Splicing, capping, tailing. These 3 processes are takes place in the eukaryotes, and due to the completion of these 3 processes, hnRNA is converted or developed into a matured mRNA. It is the fully processed hnRNA is now called as mRNA, that is transported out of the nucleus for translation in the cytoplasm. After the completion of splicing, capping, and tailing, hnRNA is called as mRNA or messenger RNA, which is transferred out of the nucleus inside the cytoplasm for translation. It is about total transcription process. Dear students, in this molecular biology unit, a number of new terms are there. So once all of you go through the textbook and you refer the textbook completion of each and every topic. Okay. So next is genetic code. Once a transcription is completed, that is processed or that is followed by genetic code. genetic code. The process of translation requires transfer of genetic information from a polymer of nucleotide to a polymer of amino acids. Is it clear? The process of translation requires transfer of genetic information from a polymer of nucleotides to a polymer of amino acids. Genetic code directs the sequence of amino acids during the synthesis of proteins. The process of translation requires transfer of genetic information from a polymer of nucleotide to a polymer of amino acid. Amino acids are building blocks to proteins. So that information is transferred from a nucleotide to a amino acid. So this genetic code, it directs the sequence of amino acids during the synthesis of proteins. The term genetic code was coined by George Gamow. The term genetic code given or coined by George Gamow. To understand how proteins are encoded began after the structure of DNA was discovered by Watson and Crick. How proteins are synthesized from the DNA. It is a great doubt or question is arised for many scientists. How proteins are synthesized from DNA. Later, George Gamow postulated that a three-letter code must be employed to encode the 20 standard amino acids used by living cells to build proteins. Amino acids are building blocks to proteins. There are 20 standard amino acids are there, and these 20 amino acids are building blocks for proteins, and a three-letter code, nothing but genetic code. This George Gamow postulated that a three-letter code, nothing but genetic code. The letters consist nitrogen bases only. They are adenine, guanine, thymine, and cytosine. Corresponds to the nucleotides found in the DNA. They organized into the codon. Codon means three nitrogen bases constitute together and it forms a codon. The collection of codons is called as genetic code.
Sarala madam.
Sir.
Conclude madam.
Sir.
Conclude.
Time అయిపోయిందా sir?
Yes madam.
Okay sir. Okay. Thank you children. Tomorrow we will discuss about genetic code as well as translation process. Thank you all of you.